So what are we going to do today? We're going to start the vision section of our perception class. And I've been telling you a lot about our vision is in our brains. But today we're going to talk about the peripheral aspect of our visual system, which of course is the eye. And what I'll try to do is link the biology and physiology of the eye to perceptual aspects, and really think about what kind of limitations this apparatus what kind of abilities as well as limitations this apparatus offers us. This is a picture of a retina. If you've been to the eye doctor and they're dilated your eyes, they're looking through the ophthalmoscope to look at the retina. They want to see that the retina is intact. This is a really nice looking retina. It doesn't have any holes or tears or anything in it. Also, you might notice that the retina has this yellow spot here. Can you guys see it? This is called the macula or the fovea. And this is where you have the highest concentration of photoreceptors, which give you the most highest acuity of your vision. We're going to talk about photoreceptors, and this is kind of like how many LEDs you have in the camera, and this is just to give you a sense of the numbers. You have about 5 million cones and 120 million rods in each of your retina. As always, I've put for you an outline of the important things for you to cover. Today's lecture is chapter 4, it's chapter 3 in the book. Thursday lecture about V1 and LGN is chapter 4, and this is why I've revised the material for the retina. Some part of the lecture today is about understanding the structure and biology of the eye, but the most important thing for you guys to remember is what is really the function of the retina. And the take-home message from today's lecture is that there's much more to the eye than just taking pictures of the world. And in fact, the first neurons in your visual systems are actually not in your brain, but actually in the retina, and these are called ganglion cells. So you already have nerve cells processing visual information in your retina. Okay, so let's start from biology. This is a cross-section of the eye. And what do we have here? So the front end of the eye is over here. There's this translucent structure called the cornea. And the cornea, behind the cornea sits another structure called the lens, which is here. And the function of both the cornea and the lens are to focus the rays of light into your eye and to provide an image of the world that's going to land at the back of the eye here on your retina. It turns out that the cornea actually provides most of the focusing power of your eye, and it provides 2/3 of the focusing power. The lens provides additional focusing powers, and really what's a big difference is that this is a fixed focusing, and this is an adjustable lens. So basically, like when you have a zoom camera, you can adjust the focus of your lens. The same thing happens here. And basically, the way it works, you see there are two muscles here that are attached to the lens, and when they push it in, it makes it shorter and it focuses the light more. And when it relaxes, when these muscles are relaxed, the lens is flatter and it provides less focusing power. Yes?
What are those muscles called?
Oh, I don't remember what's the name of these muscles, I'm sorry. But I bet it's written in the book. These muscles are very important because people who are short-sighted, oftentimes the problem is that these muscles don't contract the lens enough, they don't give you sufficient focusing power, and as a result, the image that's going to fall on your retina is going to be blurry. So people like me and some of you who have glasses, what the glass provides is additional focusing power, so we get sharp images landing here on the retina. The white part of the eye, this all this inside here, is called the sclera. And in terms of the optics of the eye, it doesn't really do much, but it's filled with a material called, it's also called the humorous chamber, and basically filled with fluid that keeps the well-being of the cell in the retina. So it's important that this thing will be in good shape, because otherwise the structure of the eye, and especially the retina, might get damaged. In terms of the optics of the eye, another structure of the eye that's important is the pupil. The pupil is actually an opening in the eye, and basically, as you all know, the pupil can get dilated or contracted, and that's adjustable by the overall light intensity. So when the overall light intensity is low, the pupil dilates, and when you go outside to a sunny room, the pupil gets contracted. And this basically works like a pinhole camera. Do you guys know what a pinhole camera is? Yes? So basically the pinhole camera controls how much light it is. If you have exactly the right amount of light, again, it provides a sharp image of the retina. If it's too wide, the image gets diffracted. It gets blurry. If it's too small, you get aliasing, and again, you can get a doubling of the image instead of it being clear. That's the pupil. Another structure of the eyes that we're all aware of is called the iris. The iris is what gives your eye its color. It can be blue or green or brown or black. But again, in terms of the optic, doesn't do much. Iris is very unique to every person. It's kind of like a fingerprint. And in fact, there's some places like in London airport that they identify people by their irises instead of by their fingerprints. Okay, so how does this work? So we have light entering our eyes. How does it form an image? And the way it forms an image is that we have a light source like the sun. It sends out rays of light. It hits objects. These objects send then photons. They hit the eye, and it actually works like a real lens. And we get an image like you did in your physics lab in high school, and you get an upside-down image of the world onto your eyes. Do you know that the images on your eyes were upside down? Doesn't bother you, huh? You don't see the world upside down. How come? Yes?
Your brain flips it.
My brain flips it. Your brain flips it. Maybe mine doesn't. No. So this is probably true. The image on the retina is upside down, but somehow in the processing of this information later on, the brain takes care of this. It knows that the images are going to be upside down, and we see the world right side up. In fact, the whole visual system is flipped in terms of the mapping of the world. Okay. We talked about the lens and the cornea, and basically the process that is important for you to understand is this process called accommodation, and this is a process that lets you see objects at different distances from you sharp all the time. So if you remember a little bit of physics from high school, if you have some object that's really far away, the way physicists like to model it is that it sends parallel rays of light into your eye, and then there is a lens there, and if you remember again something from physics, you know that the image is going to be sharp exactly at the distance that's going to be the focal length of your lens, and it turns out that the distance between the lens and the retina is exactly the focal length of our eye's lens. Okay, so images that are far away, and far away I mean like somebody like Karen who's sitting at the back of the room, something that's more than 10 meters away from me is going to be far away enough that we can model it with these parallel rays of light, is going to generate a sharp image on the retina. But of course, Manuel is sitting much closer than Karen, and I see him just as sharp as I see Karen. How does that happen? This happens by the process of accommodation. So just to illustrate my point, suppose point B is where Manuel is sitting. He's sitting close to me, not at infinity. And basically, because of the law of physics, there is a focusing power. If it wouldn't change, the distance where I would get a sharp image of point B is going to be behind my retina. And if I look at the retina, instead of having a single point of B, I will have some sort of blurry image because it will activate some range of photoreceptors on my retina. So basically, in order to see point B sharp, I need to increase the focusing power of my lens. So the image is going to be landing exactly at this distance, and I'll have only one point of light hitting my retina. And the way this works is, again, by the process of accommodation. You see these muscles here are far away from the lens. It's flat, it's relaxed. But when these muscles push in, the lens becomes fatter, and now it increases the focusing power of my lens. And now a point of light in a world will create only one point of light on the retina, and now I see it clear. What's really nice about is this process of accommodation. It happens unconsciously. You're constantly adjusting the power of your lens as you're making different eye movements in the world, and as a consequence, the world looks uniformly sharp to you. So it's basically a very fast process, and also a process that doesn't need a lot of mental power. It happens naturally. Questions so far? Okay. This is clear to everybody? So up to now, I've described the eye pretty much like a camera, only a smart camera with an adjustable lens. Yes?
You said people that are short-sighted can't contract those muscles. No?
Yes.
Short-sighted is when you can't see far, right? I just don't remember.
Yeah, short-sighted is when you can't see far.
So when you say adjusting the focusing power, would that be a relaxation of the neuron? Would that be the relaxation of the muscles? Or
No, it will be the people who are short-sighted. You might be right. Maybe I said it the other way around. It's true. Thank you for correcting me. So people who are short-sighted, the image is going to land in front of the retina, and you'll need actually a different length to bring it actually back here. Yeah. Far-sighted people. Far-sighted people will need a lens to put it back, like from B to A. And this happens usually with age, so if you've noticed, like your grandparents, they might need glasses to help them read books, and in that case, they'll need something that corrects from B to A. And this, again, becomes, with age, these muscles become tired. Yeah, you're right. Okay, so from now on, we're really going to spend most of our lecture on the structure at the back of the eye, and this is called the retina. This structure is about 0.5 millimeter thick, and basically the main function of the retina is that it transforms light or photons into an image, and this image starts to be processed, and this is the place where your retina stops being just like a camera. So I'm going to zoom into the retina and show you how it looks like under the microscope. So basically, again, the depth of the structure from here to here is about 0.5 millimeter, and it's built with layers. So basically, light enters here at the top. It passes through nerve cells that are called ganglion cells. Then it passes through a layer of cells are called bipolar, horizontal, and amacrine cells. And then it passes a layer, it gets to the back of the eye where there are cells called photoreceptors. And photoreceptors are cells that do a process called transduction, and basically that means that they turn the light energy that comes to your retina into an electrical signal such as voltage or current. So there's a little bit of an engineering oddity about the retina that if you'd be an engineer and would probably devise a device that transforms light into some electrical signals, you probably want to put the detectors up front at the top to be maximally sensitive to the light, right? So that kind of seems like a common sense thing to do. But in fact, in most animals, the photoreceptors are at the back. There are a few animals, like a couple of type of fish, where this actually happens, where the photoreceptors are at front. The retina is actually transparent, so light does go through all these layers, and the reason that it looks dark is because we've stained the cell body so we can see them under the microscope. But it still creates some problem because the way that the information gets processed, light goes here, it hits the photoreceptors, then the photoreceptors send a signal to the bipolar cells send signals to the ganglion cells. These ganglion cells now generate action potentials, and we need to send them to the brain. Now that we need to send them to the brain, there's a problem because this is the outside of the eye. Turns out that the brain is behind here. So now we have to set a whole tangle of fibers crossing through some point of the retina. So this is kind of like a design problem, an engineering problem, right? And in fact, this is what happens. So basically, the photoreceptors are in the back. They send information to the ganglion cells, and the ganglion cells send axons. These axons go to the brain through a big fiber bundle called the optical nerve. And that means that there's a point in the retina that there are no photoreceptors because you need to leave space for this fiber bundle to go out. You have a question? This, of course, is going to create a problem. Why is it going to create a problem? Yes? You have a blind spot. Why do you have a blind spot? Yeah, we have a blind spot because there are no photoreceptors. Do you know that you had a blind spot? Did you see your blind spot? You want to see your blind spot? Okay, so for those of you who don't know, and for those of you who do know, you guys have a blind spot. And I have a little demo for you. We're going to pass it around to illustrate the blind spot. And if you get this piece of paper, this is what you're supposed to do, and it will let you find the blind spot. Apparently you have a blind spot in each eye. So once you get this piece of paper, what you're going to do, you're going to close, let's say, the right eye. Once you close the right eye, you have the left eye open. With the left eye open, look at the letter L. And put it at about 3 times distance between the points, and move it back and forth so you'll see that there's going to be a point where the letter L is going to vanish. And then flip and do the same with your right eye. If it doesn't work, bring it closer to you. You have to close one eye. Did you find your blind spot? Do you have one in both eyes? Yes.
Why is it that the photoreceptors in the back is supposed
Ah. It's a very good question. That I'm asking you. So why do you think so? You have any ideas, any hypothesis? No. So we don't really know. Again, it's a design problem. It's kind of unclear why you'd put something in the back. And this is why I mentioned some fish that actually have it in the front. Some people think that it's in the back so it'll be more protected. So basically you have a layer of cells that might protect it. But it's not clear to me that this is the right answer. Yes?
So what happens if you look at a really bright light, such as that just destroy the cells?
Yeah, so first of all, somebody's asking here, what happens if you look at a very bright light? Suppose there's an eclipse and you want to look at a very bright light. Don't do that. The reason that you shouldn't do that is that what happens if you have too much photons hitting your photoreceptors, it might destroy them. Once you destroy photoreceptors, then you're going to have a blind spot in that place. So once the photoreceptor is dead, you can't see in that location in visual space anymore. So don't look directly at the sun. This is good advice. And if you damage your photoreceptors, there's no way to regenerate them back. So why do you need to close your one eye in order to find the blind spot? Yes?
Because the other eye generally compensates.
How does it compensate?
Well, like you're getting images through both eyes, so if this eye is seeing a blind spot, your other eye will fill in that spot in your brain.
Well, if it would fill in, you'd still see the letter.
No, I mean like, okay, so if you're looking with both eyes,
Yeah.
yes, you would still
Okay.
see the letter because it fills it in, but
Okay.
when you close one eye, it can't fill it in for you.
Okay. So basically, okay, so I'm just going to press you a little more. So it's true that the other eye compensates, but what does that mean about the blind side in the other eye? The blind spot in the other eye? Is it in the same location in both retina or not?
No, it's different.
In different, right? So basically, the way that the other eye compensates for it is that the blind spot is going to be shifted across the two eyes. So one eye fills the blind spot of the left eye, and the left eye fills the blind spot of the right eye. And in case, most of the time you'll get information from both eyes, but in case you have information from just one eye, then the brain takes that information that's present rather than the information that's absent. Did everybody find a blind spot? Is there anybody here who doesn't have a blind spot? Okay. So we're generally not aware of the blind spots because usually because the blind spot is not exactly in the same location in both eyes, and because of that, the image is not going to fall in corresponding points, and as a consequence, when the image of the letter R falls on your right eye blind spot, the left eye still sees it. And this is why you don't notice that you have a blind spot. But if you close one eye and now you only get information from one eye, this is when you can detect your blind spot. Does that make sense? When few people first told me that I had a blind spot, I didn't believe them. I didn't believe them because in my natural world, I always see everything, right? So you really need these illusions to show you your limitation. So this is what I mean. Our eye gives us a capability of seeing, but also it has some limitations. Okay, so we talked a little bit about the engineering design problem of the retina, and what we want to talk about are the important functions of the retina, and these are 5 functions that we're going to go and explain. And as we go and explain which functions it's happening, I also want you guys to pay attention where it's happening in the retina. I'm a neuroscientist. I like to understand mechanism and tie it with function. So this is what I'm going to go as we're going to go through the function of the retina. So these 5 functions of the retina are transduction. I'm going to explain each of them. Wavelengths encoding, light adaptation, data compression, and spatial filtering. If this sounds complicated, everything is going to become clear as we go through the lecture. So I'm going to start with this concept of transduction. Transduction is a really important function of sensory systems, and basically what it means that it transforms one form of energy into an electrical signal. In the case of visual transduction, we're transforming energy of light, which are photons that are hitting the retina, to an electrical energy, which is a change in the membrane potential. This transduction is happening in the back of the retina in the photoreceptor layers, and the most important function of the photoreceptors are to detect light photons and send an electrical signal to the next layers of the retina. How does this work? Oh, before I'm going to show you how this works, I'm going to show you a picture of your photoreceptor. So this is actually a picture of the retina, of a human retina under the microscope. And there are two kinds of photoreceptors that differ by their shape and their size. So if you can see here, these are conical shaped photoreceptors. You see they're fat and chubby, and they have conical ends. And these are called cones. They're one class of photoreceptors, and they're called cones due to their conical shape. The other class of photoreceptors is labeled here with R, and these are these skinny rod-like structures, and they're called again rods for their shape. And basically in these protrusions of these photoreceptors, in their outer segment, they contain a pigment. And a pigment, what a pigment does, it absorbs light. And another thing that happens in the pigment, this pigment is called opsin, and different photoreceptors have different variants of this pigment. For example, the pigment in rods is called rhodopsin, only it's spelled with an H. And basically what it does is that as it absorbs light, it changes its shape in a process called isomerization. So this is how it looks like in a schematic. Again, this is our, they're not drawn to scale. Rods are typically smaller than cones. This is a rod photoreceptor, and this is a cone photoreceptor, and this is an illustration of their shape and size. And basically the outer segment, which is basically this stuff here, has this pigment. And this molecule, you can see it's a very convoluted molecule. And as it absorbs photon, it changes its shape, and when it changes its shape, it changes its electrical charge, and then it generates an electrical current in the synaptic ending. So these are not neurons, they don't generate action potentials, but they'll change their membrane potential and send an electrical signal to the next layer of horizontal and bipolar cells. Now, the level of isomerization depends on the intensity of light. The more photons hit the photoreceptors, this process becomes stronger and more current will be sent out. And this will be a means for the photoreceptor to signal the overall light intensity. When there's a little bit of light, there are going to be few photons, small current. When there are a lot of photons absorbed, big current generated. So this basically, the intensity of the output, the current, would be proportional to the incident level of light. Back to your question. If you're in a dark room, you have few photons. If you go outside to the sun, especially in a sunny day like today in California, you might get bleaching of the photoreceptors. That means that the pigment absorbed as much photoreceptors as it can absorb. And what will happen if you go back into a dark room, it will have to recover back. And this time to recover back takes some time. So under normal situations, we'll have maybe bleaching of the photoreceptors, but not damage. If too much light hits them, if the intensity is too strong, like if you look straight at the sun, you might actually destroy the cell. Yes?
Sorry, what was the process called when a rod changes shape?
The rod doesn't change shape. The rod itself doesn't change shape. This molecule inside, this pigment, which is called rhodopsin, this is what changes shape. Oh, I took that slide out. Sorry. It's actually in the book. The book actually shows how this molecule changes shape. So it's only the molecule inside the rod that changes shape, and the process is called isomerization. And the book has a really nice picture of how that happens. So the outside shape doesn't change, it's only the molecule inside that changes shape. Other questions? Okay. So one function of the retina is transduction. The second function of the retina is wavelengths encoding. And again, this wavelengths encoding is still happening in our photoreceptor layer. What do I mean by wavelength encoding, and how do my photoreceptor encode wavelengths? And you also might ask yourself, wavelengths of what? So back to physics. If you remember, light is actually an electromagnetic energy, and different colors of light have different wavelengths. So if you look at the spectrum of light, this is our visible light. Do you know why that is visible to us? It's visible to us because our photoreceptors absorb light in this electromagnetic energy in these wavelengths. So if you shine electromagnetic energy at different wavelengths, like faster, higher wavelengths or slower wavelengths, it doesn't get absorbed by our photoreceptors. So the range of visual light is a range of electromagnetic energy that is transduced in your retina. And basically, this covers a spectrum from 400 nanometers, which are the short wavelengths on the purple end, till the slow, long wavelengths on the red end of the spectrum, which are in the 700 nanometer range. Okay, so light is visible to us because this is the range that our photoreceptors can transduce. And what I mean by wavelength encoding is that different photoreceptors are sensitive to different ranges of visible light. Some photoreceptors are more sensitive to the long wavelengths, and some photoreceptors are sensitive to the short wavelengths around the blue, and others are here around the green. And because we have photoreceptors that are sensitive to different wavelengths of light, this gives us a basis for color vision. So how does that relate back to our photoreceptors? It turns out that there's 3 classes of cones and 1 class of rods. So, and these 3 classes of cones are colored by the wavelengths that they're sensitive to. Their shape is the same, it's a conical shape, but they have a different pigment. Because they have a different pigment inside them, they become sensitive to different wavelengths of light. So the S cones, S goes for short wavelengths, is sensitive to the short waves around the blue. It means that its maximal absorption is of wavelengths that are centered around blue. The other class of cone are called the M cones, or for middle wavelengths, and their maximal absorption is around the green wavelengths. And the third class of photoreceptors are the L cones, and L again stands for long wavelengths, and their maximal absorption is around the red. The rods are tuned somewhere in the middle. I'll show you in a minute. And there's only 1 class of rods. So what I mean by wavelengths encoding is that different cones classes are sensitive to different colors. And this looks in a graph something like this. So I'm showing you this graph so you remember that an S cone doesn't respond only to blue, but to a range of wavelengths. It's tuned to a wave of lengths, but its maximal absorption is around blue. So again, the x-axis here is the wavelengths of the light, and I've colored it so you can see what these numbers mean. And the y-axis is the absorption range, and the higher it is, it means that it absorbs more in this wavelengths. Yes, you had a question.
I was going to ask if someone was colorblind, would that mean they lack a certain like length cone, or would it be
Very good question. So you're asking what happens in colorblind people? Do they lack one class of cones? And the answer is usually yes. So the most common color blindness is the inability to distinguish between red and green and variants of red and green like cyan and blue. And this is usually because they are lacking either the M cones or the L cones, or they have very few of them and they're not distributed evenly across the retina. It's very rare for people to lack all cones, but there is a disease called rod monochromats, and that means that these people have only rod vision and no cone vision, and they really see the world in shades of gray because they have only one class of photoreceptors. And I promised you I'd show you the absorption of rods, and you can see that rods are kind of in between the cones, so it's somewhere around the dark green range. Yes?
So to clarify, you said people with only rods would see the world in shades of gray.
Yes. They're called rod monochromats. So
The absorption of these wavelengths is just simply the detection of light or not necessarily absorption is specific wavelength.
yes. So basically, what does the absorption mean is that suppose you have a rod, it absorbs in a certain range, and as I told you, the more photons are incident, it will generate a bigger current. So basically, it's kind of like a light sensor detector. More light, more current. If you have only one class of photoreceptors, it's just going to tell you dark, less dark, bright, brightest, right? But it won't tell you what shade of color it is or what hue it is. For this, you need more than one class of photoreceptors. As humans, we have 3 photoreceptors. There are animals like lizards. They have 5 classes of cones. So they're animals that have more kinds of cones, and I want you to think what that means about their color vision as well as their visible range of colors. What happens at night? Do you guys see in color or not? No, right? When you go outside, when you look at the dark, you see in black and white. What do you think that means? Which photoreceptors are you using? Rods, right? So one other important difference between rods and cones is their sensitivity. So cones, in order to function, need sufficient level of light. For example, a lamp or some sufficient level of light outside. Rods can work in much lower light illumination. That means that less photons have to hit them in order for them to generate an electrical signal. But because we have only one class of rods, when we're using our rod vision, we cannot distinguish between colors, and this was what happens to us when we walk in the dark. The world looks to us in shades of gray. Is that clear? Yep. What happens if you have no rods? Yes, you have a question?
So animals that have more cones or rod receptors, they can see colors at night? Or?
No. So basically, animals that have more cones will have bigger spread of colors. They could see additional colors. So they could have photoreceptors that will extend maybe here or on the other side. But as far as I can tell, they're not animals that have many classes of rods. There's only one kind of rods. So what happens if your rod gets damaged? Which kind of vision will that affect? Your day vision or your night vision? Night vision, right? So if they're damaged to the rods, it's going to be hard for you to see stuff at dark. Another interesting fact about the retina is that the distribution of the photoreceptors is non-uniform across the retina. This is a really cool picture. It's a picture of the center of your retina, the fovea. It's called in biological terms the macula. And because different photoreceptors absorb different kinds of wavelengths, this image was colored by the absorption range of each class of cone. So you can see that a couple of things are visible. First of all, in the center of your retina, in your fovea, there are no rods. right? Because everything here is tiled with the colors that are absorbed by the cones. So you can see the blue ones represent the S cones, the red one represents the L cones, and the green one represents the M cones. And there isn't a gap where it's left for rods. So the center of your vision, you see sharp, you need a sufficient amount of light, and you're using your cone vision for your sharpest vision. Another thing that you might notice is that there's not a uniform tiling of the retina by different classes of photoreceptors. For example, there seem to be much more L and M cones than S cones, right? And this is pretty typical. This is typical of human retina, that there are more L and M cones than S cones. But it will vary across people if there are more L cones or more M cones. And if you look at different people's retinas, this kind of tiling will be idiosyncratic to each person's retina. Okay, so this is a homework question for you guys. So we talked about transduction, one function of the retina. Second function of the retina is wavelength encoding. That's the basis for our color vision. The third function of the retina, and you guys are going to have to read this in the book for me, is something is called light adaptation. And light adaptation is basically something about the retina that if you learn about it, it will answer the following question. And the question that you should be able to answer to me is, why can't you see well when you enter a dark room after being outside in the sunlight? And the reason I put this graph is just to orient you to which section in the book will answer this question, and it has to do with this phenomena called light adaptation and the time it takes the different photoreceptors to regenerate their pigment back to its original structure after they've been bleached by photons, for example, by strong light intensity. So I want you guys to read this in the book and figure out this process of light adaptation and understand why it might be different in its time scales across rods and cones. Okay, so the third function of the retina is light adaptation, and that means that adjusting to different levels of incident light. And instead of me telling you the answer, you guys are going to have to research this a little bit. And just to orient you again, this light adaptation process is still happening in the photoreceptor layer in the retina. So the photoreceptors are pretty cool. They do quite a lot of processing in the retina. And in fact, they also do another thing, which is called data compression. What's data compression? Data compression actually involves an interaction between the photoreceptor layer and the layer of the ganglion cells, and it is again partially a design problem. Turns out that there are a lot more photoreceptors in your retina than ganglion cells. It's a factor of about a reduction of an order of 100. So there are about on the order of 100 million photoreceptors in your retina, and on the order of 1 million ganglion cells in your retina. And the main reason that people think that there are less ganglion cells than photoreceptors is twofold. One is photoreceptors are really little. We want them to be really little because each photon is basically like a light point. So the smallest the light point is, the higher is the resolution of your vision. It's like a difference between a 1 megapixel camera and a 16 megapixel camera, right? A 16 megapixel camera has more pixels. So that means that if it's tiling the same area, the pixels need to be smaller. The smaller the pixel are, the higher the acuity. So the photoreceptors need to be small in order to give us the most accurate vision. The problem is that if you had 100 million ganglion cells, you'd need to send 100 million nerve fibers to your brain. Right? I showed you the picture of the optical nerve exiting the ganglion. So basically, each ganglion cell sends an axon out. If you had 100 times more ganglion cell, what would it mean about the size of this nerve fiber? 100 times bigger, right? So then you might have really heavy heads, right? So there's a limit. Again, it's an engineering problem. You want to have, on the one hand, the most accurate vision that you can afford. You want to achieve that by having small cells. But on the other hand, if you have too many of cells, you're going to send a lot of axons, and you're going to have a really huge nerve fiber. So there's some compensation mechanism, and this is called data compression. Yeah, you had a question? Okay, so let's see how it works. So here we're in data compression, and just to reiterate the point that I just said, so we have, again, another design problem in the retina. We have 100 million photoreceptors in the eye, but only 500,000 to 1 million ganglion cells, and the main reason is that we cannot have a ganglion cell for each photoreceptor, because that means that your optical nerve would probably be bigger than your head, or conversely, you'd need a really huge head. So basically, what the engineering of the eye does for you, it means that in order to take the original image from the photoreceptors to the ganglion cells, we need a great deal of compression. And maybe the compression has to be on a magnitude of a factor of about 100. And there are many ways, if you're an engineer and you're designing an eye, you could think about compressing data. For example, if you ever seen JPEG images on your camera, they achieve a lot of compression of the images by reducing the redundancy. So if you have, let's say, a piece of wall that is basically the same colors, they don't code every pixel in the wall, but encodes this whole area with just one number. So that's one way to achieve data compression. Another way of data compression is by convergence, and that means that several photoreceptors might converge into one ganglion cell. And a third means of data compression is to compress information differently in different parts of the visual field. Or in other words, what happens in your eye is that only part of your vision is seeing very, very sharp, and that's the central part of your gaze, your foveal vision. But the peripheral vision, your vision in the periphery, is not as sharp as the vision in your fovea. And this is what I mean by compression is accomplished by having blurry vision in the periphery. So do you guys have blurry vision in the periphery? Are you sure? Let's see. Okay, so let's see how good you are with peripheral vision. So I want you guys all to stare and pick your eyes fixated on the D. First of all, before you fixate on the D, can you see all the letters? All the letters are equally sharp to you? Okay, so let's do the following. Do what I instruct you to do. You guys think that all the letters are sharp. Fine. Now look at the letter D. Keep your eyes glued to the letter D, and without moving your eyes, see how far you can read. What are the most peripheral letters that you can read? Okay, you guys in the first row, what is the farthest letter that you can read? H. You guys are here at H. Somebody in the back row, you, what is the farthest letter that you can read? Yes, you?
Well, I saw T.
You saw all the way to T, okay. What about somebody in the middle? You could see it all the way to, oh, you saw it to this T and you saw it to the C? Okay, so basically, my first question is, nobody could see the X. Did anybody see the X? No. Okay, so why can't you see the X? You just told me before that you can read all the letters. Yes.
Because you can see the X when you look over to focus at it, but when you're only focusing on the D, it's hard to see the picture.
Okay, very good. So basically, what she said is that the reason I could see the far X in the beginning is that I scanned the letters. What does it mean I scanned the letters? Is that I moved my eyes from letter to letter. As I moved from eye to letter, I was able to focus on particular letters, right? When I focus on a particular letter, it's sharp. Because if I put the letter on my central vision, I can perfectly see it, right? But when you forced me not to move my eyes, when you told me to stick it on a certain letter, this is when I noticed that I can't see well in the periphery. Okay? So again, in normal situation, when I look at you, when somebody told me the first time, you see blur in the periphery, it seemed really odd to me, because to me the world seems sharp equally. As I look at it, it doesn't look like parts of the world are blurry. It looks to me equally sharp. The reason it looks equally sharp to us is that we move our eyes. As we move our eyes and we scan the visual scene, different parts of the scene fall on our foveal vision and we see it sharp. But if we fixate our eyes, it's just then we notice that we cannot see things very clearly in the periphery, and our first instinct is to see something in the periphery is to move our eyes so we can see it clearly, right? And this is just to illustrate what I mean by data compression or blurry vision in the periphery, and when you need something like reading that needs very accurate vision, you can see that you cannot see very well into the periphery. Why was it different for the people in the front and the people in the back? Why the people in the front could see less letters than people in the back? Do these guys need to go see the eye doctor? No. So guys, help me. What's the difference between people in the back and people in the front? Yes.
The periphery is smaller in the back.
So what falls in the periphery are farther away letters, yes. So the periphery is not smaller in the back, it's equal periphery, but what happens is that the visual angle is different between people in the back and in the front, and basically more letters are going to fall in the foveal region in the back than in the front, and I haven't made these letters too small for people in the back not to be able to resolve them. That makes sense? So don't worry, everybody has here good vision, but the visual angle is going to be dependent on how far away you are from the screen. Okay, so how is this data compression achieved, again, biologically in the eye? And I told you I'm going to link between a mechanism and a percept, so this is another example. The way it's achieved is that you have non-uniform sized photoreceptor as well as non-unified sizes of dendritic arbors of ganglion cells between the center of your vision and your peripheral vision. The left panel shows the tiling of the photoreceptors in the fovea. Each photoreceptor here is a rod or a cone. Cone, right? In the fovea, only cones, okay? So you can see that they're very small, okay? Each photoreceptor is small, and a photon really needs to hit exactly this point in order for that photoreceptors to generate an electrical current. If you look at the periphery, you know what these big globs are? These are cones. So just compare the size of the cone at the fovea versus a cone in the periphery, and you can see that it's much bigger. That means that the spatial acuity is going to be lower, because if a photon hits here, or here, or here, or here, it doesn't matter where on the photoreceptor area, it's going to generate a current. And this is why you have a bigger tiling, you have a lesser visual acuity. The other thing you should notice is that your rods are still pretty small in the periphery, and this is a general finding that in a given visual angle, how far away you are from the fovea, cones tend to be larger than rods. Okay, so the data compression is achieved biologically by having a non-uniform tiling of the retina with different size photoreceptors, and this is summarized on the plot on the bottom. So this is distance from the fovea in the retina. This is a cross section, and this is the density of cells. The higher the density the cells is, that means that they're smaller in size, and you can see that these are different observers, but for each of these retina we have a decline in the density of cells as you extend to the periphery, and this means that for everybody there's going to be blurry vision in the periphery. Questions? So a lot of facts this lecture. Just go over this and also read the book. Okay, so now we're going to talk about the last function of the retina, and that last function is called spatial filtering. We're going to talk about it twice. We're going to talk about it today, and we're also going to do some retina mass in the sections this week, which are going to be led by Christian. So one of the things that you should remember is that your retina does some mass for you, and we call it in engineering spatial filtering. And this is happening now no longer in the photoreceptor of your retina, but in the neurons called ganglion cells. And these neurons generate action potentials. So if you put an electrode next to a ganglion cells, you could actually measure spikes. And this is the output of the retina is sent from the ganglion cells to the brain through the optical nerve. What do I mean by spatial filtering? Okay, I'm going to show you an illusion. This illusion is called simultaneous contrast. And basically, the question is the following. Which of these squares, this square or this square is darker? You guys are whispering. How many people think that the left square is darker than the right square? Some of you. How many people think that this square is darker than this square? You think that this one is darker, this one, than this one? I'm tricking you again. Yes, you don't want to answer. You want to know what's the right answer. So, okay, let me ask it in a way that you won't feel that I'm tricking you. Which of these squares appears darker? Would you agree with me that the left one appears darker? Okay, thank you. Of course I'm tricking you. Okay, you want to see that I'm tricking you? I took away the background. What's happening? Which one is darker? They're the same, right? I'm going to put it back. Oh. I'm going to put it back. The one on the left appears to be darker, right? I take it away. You can see that these are the same darkness, right? I just copied it. Okay, so it's actually not me tricking you. It's probably your retina tricking you. And this is what I mean by spatial filtering. So, one of the findings about the retina is that ganglion cells have a particular structure. I'm going to talk to you about the structure. And basically, because of the structure, it leads to certain kinds of visual illusions. And this is what we're going to do a little bit now and more during the sections of this week. And it turns out that the output of the ganglion cell is dependent not only on how much light hits its center, but also how much light hits its surround. And it has this effect that if the surround is brighter, it inhibits the center and will make it appear brighter. If the surround is dark, it will inhibit less the center and it will consequently appear brighter. This is called in the book lateral inhibition. So basically, what this means perceptually is that already there's some processing happening in the retina that what you perceive at a certain point is determined not only by the light from that point, but also on the light in its surround. This illusion is called simultaneous contrast, and during the section we'll show you a couple of other illusions, maybe Mach-bands, maybe the Hermann grid, that are thought to be because of the spatial filtering in the retina. So what I particularly like about the retina, and I particularly about these kind of illusions, is a lot of illusions that we don't really know how they work. And probably this illusion also, we can only partially explain it with the mechanism of lateral inhibition. But at least this gives us a good anchor to start linking between physiological mechanisms and perception in a very clear and mathematical way. So it's not me tricking you, it's your retinas. Okay, so this is a picture of a receptive field of a retina. It's not of a retina, sorry, of a ganglion cell in the retina. The person who discovered the function of these cells is a professor named Stephen Kuffler, and he was working in the early 50s in the Harvard Neuroscience Lab. This is a picture of Kuffler, and this is actually a reprint from his Journal of Neurophysiology paper from 1953. If you believe it or not, they actually drew by hand the results. So it seems impossible in today's world, but science has evolved. So this is actually an illustration that he hand drew, and this is a result of his experiment. What did he do in the experiment? He took anesthetized cats and put electrodes in the retina, listening to the ganglion cells. So even though the cat is anesthetized, the retina, the ganglion cells are still active. And basically, what he did, he shined different spots of lights onto the retina and wanted to see where he shines light, what would be the output of the cell. And again, when we're measuring electrophysiology, the more what we want to see is some sort of stimulus that modulates the firing rate of a neuron. And then he coins this term called receptive field. What is a receptive field? A receptive field is a place in the world, in visual space, that if Kuffler shines light in that point of space, then you get a modulation, either an increase or a decrease in the firing rate in the ganglion cell. So basically, it looks like this. So if he shines light in point B, this is like the world. Suppose the cat is staring at a screen, and he's recording from the ganglion cells. If he shines a point of light in B, we get an increase in the firing rate of the neuron. This is called the excitatory part of the receptive field, and it's indicated by the plus sign. If he shines light at the surround, that's indicated by C. Even if though he's shining light, he's measuring from a ganglion cell in the retina, he's actually getting a decrease in the firing rate of that neuron. And this is called the inhibitory surround of that ganglion cell. And in the books, it's schematized as a circle, but in his original paper, basically, these things are often kind of oval, and basically this shows the excitatory center and the inhibitory surround, and this is basically 1 millimeter. This is the size in the region in the retina that gets activated by this point of light. So it's a little bit confusing, this notion of receptive field, but often when we talk about properties of neurons in the visual system, we're interested in mapping their receptive fields or understanding what kind of stimulus put in which part of visual space will modulate their response. So I have only a couple of minutes left, but I would like to show you the experiment that he did. So this is basically Kufler's experiment. He's putting in an anesthetized cat. The cat is in a dark room. He's measuring signals from a ganglion cell. And this is an example of what I call a center-on, surround-off receptive field. Even though the cat is in darkness, you will measure some action potentials from that neuron. You guys remember how this is called? Baseline firing or spontaneous firing, right? So the neurons are never silent. Even in the absence of a visual stimulus, there are going to be some action potentials. Then he's shining in a particular point in space, a point of light, and you can see the red time indicates the duration that the light is on. So this is a time axis, no light. He shines a spot of light, he takes it off, and you can see that there is an increase in the firing rate of the neuron, and this is because the light is hitting its excitatory center. The interesting thing that happened when he shined a larger spot of light. The intuition would be that if you shine more light, you might get even more action potentials, right? Turns out, just the opposite had happened. So even though he's shining a bigger light spot, you actually get less action potentials than if you shine a small light spot. And this really puzzled Kufler for a very long time. How could it be that he's shining, it's a visual system, you're shining more light, you're getting less activity. So what he did, he took the difference between these two lights and shined a disk. So when he shines a disk of light, there's no light in the center, he's getting actually something called inhibition. So he's actually getting less action potentials than the baseline firing. So basically what's happening is that when you're shining light to the excitatory center, you get an increase in the firing rate, an excitatory response. When you're shining light on the surround, you're getting an inhibition of the response. And actually it's when you turn the light off that you might get action potentials, like in this case. How does it relate to our phenomena of center surround? Suppose you had a ganglion cell that was centered on the left square. This is not drawn to size, but let's just suppose for the context of the example, just so you get this idea of surround suppression. If you have a ganglion cell that this is its receptive field centered just this way, the excitatory center will get some light, but it will get a lot of inhibition from the bright light on the surround. In contrast, if you have another ganglion cell that was centered on the left square, it gets exactly the same excitation in the middle because I made the squares exactly the same, but it gets less inhibition from the surround because there's more light in the surround. And as a consequence, because there's more inhibition on the surround, which is when it's hit with a brighter spot of light, you get this phenomena of simultaneous contrast that an equally gray square, when it's surrounded by a lighter patch, appears to be darker than when it's surrounded by a darker patch because it's getting more inhibition from its surround. Okay? This is complicated. We're going to repeat this in the section, and you guys are going to do some retina math for me. Okay? Thank you, guys.